In this video, I'll take you through example 2 on the Pipebio documentation site, analysis of paired VH-VL sequences. You will learn how to import FASTA files, annotate sequences, and pair heavy and light chains from the same molecules. The example data is a set of DNA sequences from 137 antibodies in advanced clinical stages. This is a public dataset first published by Jain et al. In 2019. To start, download the raw FASTA files from the Pipebio documentation page to your local machine. Unzip the downloaded files and inspect the contents. If you are on a Windows machine, make sure you extract all the contents fully. There are two FASTA files present in the folder. One has the heavy chain sequences, the other has the light chain sequences. Note that the heavy and light sequences share a common unique identifier. In this case, the plate ID and the well number and the antibody name. We will use this information later to pair the corresponding sequences. It is also possible to have both heavy and light sequences in the same FASTA file, as long as there is a unique identifier that pairs the sequences together. In Pipebio, create a new folder in the project. You can import the files either by going to File, Import Files, and browsing to the location on your local machine, or you can simply drag and drop the files directly into the new folder. If metadata columns have been configured for your organization, you'll see an extra dialog to set those values. If not, you'll proceed directly to upload. Within a few seconds, the sequences from each FASTA file will be in separate documents. Open the documents and inspect the sequences. Note how the name columns contain the unique identifier that helps us to pair those heavy and light sequences. We can annotate the sequences to identify heavy and light chains. Select both documents, go to the sequence menu at the top, and choose Annotate. Choose the IgG scaffold, and choose the latest version of the human germline database. Finally, click Annotate. Open the annotated sequence documents. Use the status chart to get a quick overview of the annotation results. All sequences were annotated correctly, and the scaffold detected just a few sequence-based liabilities in the sequences. You can check if heavy and light chains have been correctly identified by inspecting the chain column. For the light sequences, all sequences should have a VL annotation. For heavy sequences, all sequences should have a VH annotation. We compare the annotated heavy and light sequences using their unique identifier and the chain information that was generated in the annotation step. Select the annotated sequences documents. Go to the sequence menu at the top and choose Pair Annotated Chains. For the Pair By option, choose the name column. The chain column should automatically be selected for the chain option. Click Pair Chains. One output document will be produced, containing the paired sequences for each antibody. Each row is a different antibody. In the sequence viewer, you can toggle between the heavy and the light sequences using the drop-down above the viewer. Notice the chain column is now VH-VL. The paired sequences can now be used as input for downstream tasks, such as clustering.